the last enzyme, pyruvate kinase. What this pyruvate kinase do is, it is going to convert this phosphoenol pyruvate into pyruvate. Phosphoenol pyruvate is converted to pyruvate, done by pyruvate kinase, and you are breaking the phosphate attached to this second carbon here and transfer that to ADP. Phosphate is transferred to ADP, and that's how you make ATP. So this is again an example for substrate level phosphorylation done by pyruvate kinase. Okay, so far we have two examples for substrate level phosphorylation in glycolysis. One is phosphoglycerate kinase, when 1,3-bis phosphoglycerate is converted to 3-phosphoglycerate. The second one is this one, phosphoenol pyruvate to pyruvate. Again, you are going to make ATPs. So 2 molecules of phosphoenol pyruvate, 2 molecules of ATP for every molecule of glucose. Remember that 2-2-2 thing, right? So this is how glucose is oxidized all the way to 2 molecules of pyruvate and making ATPs. So initially we have spent 2 ATPs, later we have made 4 ATPs. So 4 minus 2 will be net 2 ATP. And that net 2 number will come from this reaction. Is it not? Initially you spent 2 ATPs, and in the phosphoglycerate kinase step you got back 2 ATPs, so equalized. The net gain is there in this reaction. Is it not? So what if this enzyme is down? No ATPs, because this is the step where it was getting net ATP, like red blood cell. It was getting net ATP at this step, and you are blocking that enzyme. So that means your red blood cell won't be able to generate sufficient ATPs. So if the red blood cell is not able to generate sufficient ATPs because this pyruvate kinase enzyme is deficient, pyruvate kinase enzyme deficiency. So this enzyme is, remember, what I have written is deficiency, not complete absence. Deficiency, maybe instead of 100%, there is a 50%, 40%, 60%, something like that, right? So pyruvate kinase enzyme deficient means there will be decrease in ATP. When there is a decrease in ATP, remember, especially in the red blood cell, other cells are fine. Red blood cell, which doesn't have mitochondria, it is entirely dependent on anaerobic glycolysis. Remember anaerobic glycolysis? So red blood cells are heavily dependent on glycolysis, anaerobic glycolysis for ATP generation. And the last enzyme was the one that was giving that ATP, and that is down now. So overall, there will be drop in the ATP levels in the red blood cell. Once that happens, what will happen to the pumps, active pumps that are present over red blood cell membrane? The most important active pump that you know is sodium-potassium pump. The sodium-potassium pump, it is means maintaining that sodium-potassium level in and out. So this pump will fail. When the sodium-potassium pump fails, what will happen to the sodium inside the cell? It just stays inside. It is not pumping out. So sodium stay inside. Once the sodium stays inside, it's going to drag chloride. Chloride comes in. So you have sodium chloride, salt. And this sodium chloride, what it does? It will bring water in. And that means your red blood cell will swell. And as it swells, it will break open. And that causes hemolysis, leading to loss of hemoglobin from the red blood cell. And that causes hemolytic anemia. Anemia means what? Decrease in hemoglobin, right? So the pyruvate kinase deficiency causes hemolytic anemia. It is the most common glycolytic enzyme. If you are only restricting yourself with glycolysis pathway, this is the most common enzyme leading to hemolytic anemia. Okay, so it is an autosomal recessive condition. Pyruvate kinase deficiency follow autosomal recessive inheritance pattern. That is important because there is another condition that you are going to study where there will be hemolytic anemia. In fact, that is the most common considering every metabolic pathway. If you consider all metabolic pathways and see which is the most common cause of hemolytic anemia in terms of enzyme deficiency, that enzyme is glucose 6-phosphate dehydrogenase enzyme. We are going to study G6PD deficiency, the most common out of all metabolic pathways leading to hemolytic anemia. Whereas if you restrict yourself with glycolysis, pyruvate kinase is the most common. Anyway, not really that important. Just know that pyruvate kinase enzyme deficiency leads to hemolytic anemia. And it is autosomal recessive. Why this is important here? Although many diseases are autosomal recessive. You know, enzyme deficiency diseases, commonly they are autosomal recessive only. So that means we don't need to worry too much about it, as long as you know autosomal dominant and X-linked conditions. Whereas here, it makes a difference. Why? Because the most common cause of hemolytic anemia that I said is G6PD, glucose 6-phosphate dehydrogenase deficiency. So G6PD deficiency. G6PD deficiency is most common cause of hemolytic anemia. And this G6PD deficiency, it follows X-linked inheritance pattern. X-linked. X-linked means what are the, who will be symptomatic? Only males. Males are symptomatic, females are carriers. And that is what you see in G6PD deficiency. Whereas pyruvate kinase deficiency, autosomal recessive, that means both males and females are affected in the family. And that information will be given in the stem. A patient so and so, they will explain, and then upon a family history, his sister also having a similar condition. You're saying a 22-year-old boy, and you're looking at, and then his sister also had similar issues, both male and female in the family. At that time, and the case stem is about hemolytic anemia. At that time, you should not go for G6PD. Go for pyruvate kinase, because both are affected in the family, both males and females. So whenever you see males and females affected in the family, you should rule out X-linked condition G6PD. Be with pyruvate kinase. And also you'll see right shift in the oxygen dissociation curve, because 2,3-BP is increased in pyruvate kinase deficiency. And one more thing, in pyruvate kinase deficiency, you do not see something called hinge bodies. Hinge bodies. You don't see this. No hinge bodies. Why I am written, means I have written here something which is negative sign. Something which is not there, why should we write? There are so many things not there, I didn't write. But I have written only hinge bodies, no hinge bodies. Why it is important? Because G6PD deficiency patients will show hinge bodies. So that's how you are going to differentiate. These are two, and G6PD deficiency patients, 2,3-BPG is not significantly increased. 2,3-BPG not significantly increased. That is why these points will help you in differentiating between pyruvate kinase and G6PD deficiency. That is where you will be in the exam, between two choices. Let me be honest with you. When you see hemolytic anemia case, in block 1, I teach both glycolysis and pentose phosphate pathway. That enzyme comes in pentose phosphate pathway. That's where you see G6PD. And when you look at the case stem, it's a hemolytic anemia and so many things you are seeing. And in the choice, you see both pyruvate kinase and G6PD. Both of them are there in the choice. And you will be between only those two choices. Other three choices you will rule out. But you are between those two. Which one to go for? Because you will have both the questions asked. Later you will get the other question also. There also you will see pyruvate kinase as a choice, distractor. How do you rule out? Those are the points. Look for if there is increase in 2,3-BPG. If it is yes, think about pyruvate kinase. Look for if there is hinge bodies. If there is no, think about pyruvate kinase. See if both males and females affected in the family. If it is yes, pyruvate kinase. Like that. So if you answer other things this side, go for X-linked. G6PD. If the only males are affected, females are fine, Hinge bodies are present, there is no significant rise in 2,3-BPG, hemolytic anemia is there, that's G6PD. So this is how it will help you in differentiating between two conditions. So make sure you know that part as well. So that's about pyruvate kinase deficiency. Any question before we go for break? Any question? Yes. Okay. Hinge bodies, these are precipitated hemoglobin molecules in the red blood cells. I'm going to explain you more on this in G6PD deficiency. But at this point in time, I'll just let you know that Hinge bodies are, these are precipitated hemoglobins, okay, which you don't see in pyruvate kinase deficiency.